And again, after like once you like learn to read, like it just isn't like a thing that we talk about really. So, like I said, reading academic papers is hard. And it's hard for a whole variety of reasons, but like just to go over a couple of them, right? Like, so there's technical jargon. Like they use like very particular words that like we don't use in like everyday speech, and that makes it like difficult to parse what they're saying, and sometimes it makes it like really difficult to comprehend what they're saying. Additionally, like when we're reading scientific papers, like they're new and groundbreaking and novel, and that's why they're being published. But that means that they're like frequently built upon like numerous advanced concepts, like not things that like you're taught in like textbooks, not like your basic fundamental things that like we all have like mastered at this point. It's like like they are built in cutting edge upon like just below the cutting edge. And so like these papers are built on the shoulders of giants. It requires a lot of prior knowledge and a lot of understanding of like the minutia for like everything to really like delve into what the paper is trying to say. And then finally, the papers that get published are the ones that are noteworthy. And as a result, they tend to provide information that is like contrary to the expectations. So like that's why they're noteworthy. That's why they're being published. And that can cause some cognitive dissonance where you're trying to hold two different ideas in your head at the same time, and you just end up getting confused because you're like, but I thought this way, but they're saying this way. Which way is it? And it's like, well, like there's just a lot of nuance in understanding what they're trying to say. And to kind of get back to like the difference in in this information. So no one teaches us how to read academic papers. So like say we'll just kind of flow through, like throw forwards in time. So like someone makes a discovery and they write a paper about it. And then there are enough discoveries about that thing that someone decides to compile a bunch of that stuff and do like a review paper. And then there are like, and then that enough people kind of know about that subject that they begin to add like footnotes and things into textbooks and like regular documentation. And then eventually like it reaches a critical mass and it gets added into textbooks. And unfortunately, for like most of our career, we have very, very little experience and just with like reading papers and reading review papers and looking at footnotes, and we spend so much of our time just looking at textbooks. And then we have this double whammy where the most difficult group to understand, like the one that requires like the most nuance and understanding, is the one that we have the least exposure to. So of course reading papers is hard. Like it's entirely different skill sets than what we've been using for most of our time reading textbooks and understanding textbooks. And it's just there's a lot of information, and it's very nuanced. They're talking about like minutia, very fine points, very fine granular points that just are largely overlooked in textbooks because they're too large and they're too granular. Textbooks want to make like generalized statements and generalized trends, and papers don't want to do that. They want to make very specific claims, and taking all of that together is taxing. It's hard. It's difficult. And so what I kind of want to do today is we're gonna we'll get around to the paper if we have time. But I think more importantly, let's go over this. How do we read academic papers? How do we dissect? Like, how do we let's develop a process to read through papers and get information and like get understanding and get information out of it and make that information meaningful. So that's what I'm actually going to be presenting today. Mostly. All right. So first and foremost, like I want you guys to understand like information processing is a skill. And it's a skill that we've all developed to some degree. Like you guys are, let's say, we're able to like go about our lives safely, I'll say all the time without thinking about it, because there's a level of information that we're always intaking from our environment, and we're very used to processing it. We're very comfortable with processing it. So I'm gonna define these terms before I go any further. So information is just any kind of stimulus. So visual, auditory, conceptual, tactile, or miscellaneous, that relays something of meaning. So again, we take in a lot of information all of the time. Right now, you're taking in information about the sound of my voice and the way that I'm saying things and my facial expressions. You're also taking in what is on the page, and you're reading the page, and each part of the page has a particular word, and like you're putting those words into context, and you get where I'm going with this. My point is that there's a whole bunch. You guys already have a lot of these skills. We just have to take those things and we apply them in like under a different setting. So these aren't like any like formal terms. These are like things that like I am coining like for purpose of this discussion, and I just think that like they make sense. So like this information limit is the amount of information that can be meaningfully inhaled at a given time. So like everyone has an individual information limit, and that will vary, from individual to individual, from time to time, what's, in setting to setting, in context to context. But there's a theoretical cap, which is what I'm going to call this information horizon. And that is the amount of information that can theoretically be obtained by a given stimulus. If something is hot, like there's only so much information you can receive about the temperature of an object. Okay, so we're going to do a little experiment here. I'm going to give you guys 10 seconds to look through this picture, and then I want you to tell me what's going on in the picture. Yes, some of you would be like, you want to just spend some time watching, to take your amount of time you've watched League of Legends and make it relevant. And I'm like, well, yes, but no. All right. So what was going on in the picture, guys? Feel free to speak up. Guys, girls, team, what's going on in the picture? There are no wrong answers.
There were, I guess it was showcasing two players who were.
All right, Anna says a League of Legends battle.
I'm sorry?
Sorry, I'm just reading the chat. Anna said it's a League of Legends battle. Does anybody else have anything else?
Team blue and red.
They were showing two players, I guess, who were, I guess, doing something, completing some objective in the game.
Strategy game.
I think was it red team or blue team? One of them were defending the tower, and another one was attacking. They were almost there. I don't know the terminology how they use in the game.
Blue team's defending turret. Yep.
Yeah.
Yeah. All right. So let's see. Anyone else have any other observations that they made they want to talk about? All right. So I did that same thing, and I'll just kind of go over the things that I noticed. So like Anna said, it's a League of Legends game. Specifically, it's a competitive LEC game between Fnatic and SK Gaming, and it's about best of 5 series. SK Gaming is up in kills, but Fnatic is up in gold. Fnatic has one more tower than SK Gaming. Both teams are tied in with the number of drakes. One of them has earth and one of them has air, but I couldn't remember which one was which, so I just wrote it. And they're just quite at about the 15-minute mark, which means we're transitioning into the mid game. The Fire Drake spawns in 3 minutes. Baron spawns in 5 minutes. The Rift is going to be an Inferno Rift, which is going to open up more jungle routes, which is actually going to favor SK Gaming a little bit. Fnatic has a mid-game composition with with 5 different champions. SK Gaming has like an early mid-game composition, and they've got 5 champions. SK Gaming won the top lane, but really hasn't been able to shut out the opponents. Fnatic is winning mid lane, and both players on the bottom are kind of even. And overall, like, it looks like Fnatic's going to win this game. I got all of that in that same amount of 10 seconds. So is this impressive? Well, no, not really. I've just spent a lot of time learning about this game and how it works and all of the different minutia of it. And so when I look at it, I'm looking for very different things than what you guys are looking at, because you guys are only looking at broad picture stuff. So the point of this So different people get very different information from that picture. And so that information our information limit can be expanded to approach that information horizon. Like, as much as I was able to get off of there are people who are able to get a lot more out of that same pieces of information. Just because they've spent even more time than I have. They have a better comprehension of it than I do. But my point is that we can expand this by doing these things. So we can have more frequent exposure to the material. So in this example, you could watch more League of Legends games. Higher duration exposure, you could study that picture longer, and the longer that you study the picture, maybe you would learn more things, or you'd pick up on more things. You could decompose the elements, of the picture into more basic principles. So like you guys said, we observed it was a League of Legends game, but how does a League of Legends game work? How do you know who is, how do you know who is winning? How do you know that it's an early game composition or a mid game composition? How do you know any of that? Well, you just decompose it into the smaller elements and then look at them individually, and then as you gain experience, you can begin to put them together. So we can form models of the information. Again, that same idea of we're going to take these small pieces of digestible information and we're going to put them together into a larger context. We can begin to do critical analysis of our process. What were we drawn to? What were we paying attention to? Is that really the thing that we should have been paying attention to? And then finally, just repetition and practice. Just do this type of thing over and over and over again. And the more that you do it, the better you will be at it. So what does a League of Legends snapshot have to do with understanding papers? Probably what you're all thinking at this point. You just wanted to sit here and talk, and gloat about your League of Legends knowledge. Oh, okay. So my point is that the principles that you use for analyzing the data is the same. They're the same. They're just, you need to apply, they're just in different contexts. So you apply them to different, if you apply them to different things, you're performing the same actions. So we can break down complex information into smaller and more easily digestible chunks. And hopefully those are more meaningful than the complicated thing. We can focus on particular areas to try to build up our knowledge base. You don't need to understand the whole paper the first time you read it. If you can just take away a few key portions, it's fine. There's nothing wrong with that. We can read more papers more frequently. And then we can engage in practices like this, where we examine what we do, why we do it, and is there a better way of doing it? It's just some, I say, like the same basic principles that we just described, we can apply to a different situation. So with that in mind, let's kind of start getting into the paper format. So when you do a paper, it tends to be broken down into like 5 distinct sections, depending on like what the publication is. Like sometimes they have more, or they have different sections, or they're in a different order, but largely like this is kind of the general overview. So you have an abstract, which is brief technical summary of the paper. I say brief in quotes because we all know that abstracts vary quite a bit. So then they have the introduction, which is like an overview and a history of the problem. We have the materials and methods, which is the list of the objects and the techniques used for said problem. We have the results and the discussion, which is just the list of the results from the experiments and then analysis of said results. And then the conclusion, and the conclusion of the synopsis of the claims and the evidence presented in the paper. And then we have, a couple of other things, which is like the figures, tables, and legends, and like the supplementary material. And those are, all papers will have figures, tables, and legends. Some of them will have supplementary material. All right. So when you first read a paper and are going through it, don't say only focus on these 3 areas. So read the abstract so that you kind of know what the paper is going to be about. Read the conclusion so that you understand what the authors thought that they did, and then go through each figure, each table, each legend, and try to understand what is being relayed to you in that, in that figure. As you know, I put as necessary, you can refer to the materials and methods. Just I don't understand what's going on in this figure. I'll look into so then we look into the materials and methods to see how they made that figure and the experiment that they did to perform it. So that you can begin to, so you can kind of begin to unpack what's being laid in that figure. Warning, most figures are dense, and they're dense intent, they're trying to relay as much information to you in as little, in as small, a graphic as possible. And so this can take some time, and it's again, it's like all of this, it's hard. Right. So again, so this is what we're trying to, these are the questions we should be asking ourselves as we're reading through all of these portions. So the abstract, what is this paper about? What is it trying to say? When we read and as you read it, you need to be able to answer those questions. If you can't answer these questions, reread it. Conclusion, what does the paper claim? What questions does the paper claim to answer? For the figures, tables, and legends, how does the paper address the subject, and what support does this offer for its claims? So after we we need to conceptually understand what's going on in each figure and begin to make connections on how it ties back to what they're saying in their conclusion section. And again, if we have to go over the materials and methods, what do the authors do to identify the evidence to support their claims? Does it stand up to a reasonable amount of scrutiny? What about an unreasonable amount? Questions are the ones you want to be thinking to yourself. You don't have to have answers to them immediately. All right. So however, we do need to repeat this exercise until we can independently explain the following things. So we need to be able to clearly state what the paper is attempting to do. So basically summarize in your own words the abstract and the conclusion. You should be able to provide a general explanation for what each figure and subfigure is illustrating and how it relates to the purpose of the paper, at least on a conceptual level. So they have this graph. This graph shows A comparison to B. The values on A are much larger than the values on B. Therefore, it shows that A is better at performing some task, and that relates to their conclusion that A is a good treatment for this disease. Pretty very, very basic, fundamental, first principles observations. And then you should have some idea as to how those figures were created. And if you can't intuit how that figure was created, you can read the legend and then search out the corresponding section from the materials and methods. Cool. We have any questions on the idea for the first read? Great. So this first read shouldn't take this doesn't sound very imposing. This doesn't sound like it's going to take an hour of my life. It's just going to be I can spend 15 minutes, look through some pictures, and explain how those pictures relate to their claim. That doesn't sound too terrible, right? Much more approachable than read this 37-page paper and be overwhelmed the whole time. All right. So again, don't worry if you have to repeat this 15-minute exercise a couple of times to find it, to really grasp what's going on. As you get better at doing this, as you do this more times, as you practice, you'll get better at doing this. All right. So for the secondary exposure, we want to focus on putting the various pieces together. So in the first iteration, we wanted to conceptually understand how all these things tied into what they were claiming. The second iteration, we really want to put figure A together with figure B to understand how those interactions work. So in order to do that, we're now going to sit down and we're going to read the results in the discussion portion. And like I said, we're searching for connections between each figure and table. And if we're ever confused, we can just pause and read the corresponding portions of the materials and methods whenever necessary. All right, and then once we're done with that, then we get to the final iteration. We want to critically examine the paper, and we're looking for extensions and weaknesses. So we want to read the introduction. We want to thoroughly understand that what problem the paper is trying to address. Then we want to read all of the materials and methods, including all of the ones that we haven't looked at before. Here we want to critically examine their methodology, and this may require some e-search. I recommend DuckDuckGo. We find a term that we don't recognize, or they're spending a lot of time talking about this gene or this pathway that I don't really know. I'll just look it up. I'll just say, and I'm okay. Okay, now I understand. They're talking about this particular metabolic pathway, and it takes this thing and it does that thing, and those things play different roles in this thing. All right, I now kind of conceptually understand what they're talking about. We now want to re-examine the figures and the tables. We want to reread the results and discussions, and this time we're going to look for leaps and generalizations. You claim this, but is that really, when I look at that data, is that really the way that I interpret it? Is that the only valid interpretation? Is there something else is there another equally valid interpretation? And then reread the conclusion, and are you convinced that their claims are correct, that there is no other alternative, or that every other alternative has been proven wrong. So this 3-step procedure should make reading papers less overwhelming and more enjoyable. So I'm just gonna reiterate, go over those things again. So in the first read, we're focusing on the abstract, the conclusion, and the figures. We want to get high-level concepts, basic understandings of the paper, and we are reading to conceptualize. We're not, let's say, it doesn't matter that there's a whole bunch of stuff that we haven't read before or that we're unfamiliar with these things. We're just trying to conceptually understand what they're doing and, let's say, and how those things relate. On the second read, we're going to go over through the results and the discussion, and we're going to try to put these concepts together. We're going to try to formulate and understand the presented argument. So this time we are reading to understand. And then finally, on the third read, we're going to include the introduction, the materials and methods, and we're going to critically examine each aspect of the paper. We're going to be reading to evaluate, and it's a bit of a like self-check, like, are you really there? To quote one of my mentors from undergrad, if you don't have any questions at the end of the paper, you're not there yet. All right. So again, just a reminder, science is hard. Every paper, the authors, like we've all seen the papers with like lists of authors, right? In each of, say, in every paper, those lists of authors have individually put tens to hundreds of hours into each paper. So you can't expect that you're going to just have high level or perfect comprehension without some work. And that like the amount of work, will change over time depending upon like what the subject of the paper is and your experience with that subject. And then, just a reminder that as you develop more diverse and comprehensive background, reading the papers will just become easier. You will be more familiar with the plots and the methodologies. So like analyzing the different figures will just become easier. You won't have, you'll have an increased vocabulary and familiarity with jargon. So less words will trip you up. You'll know what they're talking about, and you will be doing less e-searching. And then you'll have essay, and then broadly, like knowing one thing very well means that you can make associations with other topics. So as you become an expert in a particular area, just understanding other areas just becomes easier because you have a framework that you can just map everything else to. All right. So now we've reached about the halfway point of this of the talk. So like, are there any questions over like how I'm suggesting to read papers? Again, feel free to speak out or just use the chat. So methodology, sorry, so Pradeep's asking about methodology papers. And method papers are, I still think you're going to use something like this approach, but each section will be a little bit more involved, because like the point of the methodology paper is they're trying to sell you on this new method of doing things. And so largely they don't like, their results are, we performed this operation and it worked, or we believe that it worked. And that just, if that doesn't quite match, you're right, that doesn't quite map so nicely to this, but just you're going to spend a lot of your time here in the here in the read. And at that point, like you maybe need to include, you'll just read more of the materials and methods as you go along. That kind of answer that question, your question, Pradeep? Excellent. Okay. Any other questions? Any other comments? Does reading papers sound less like a Sisyphean task? Oh. All right. So with that out of the way, let's say, so now let's get back to what the the subject of the presentation. Like, let's go over like this RNA targeting with CRISPR Cas9. Right. So first and foremost, let's talk about CRISPR Cas9. So CRISPR Cas9 is this is a bacterial protein. It's part of like their like an equivalent to the immune system, like a bacterial immune system, where it takes let's say this sgRNA. So it takes a guide RNA. We'll say, and the CRISPR-Cas protein identifies this loop here, and then will bind to it. And then once it's bound to this guide RNA, it will then look around any DNA that it finds. And it's looking for what's called the PAM sequence. And every iteration of Cas9, like different bacteria, have a different, not all of them, but most of them have different variations on PAM sequences. So PAM is just a abbreviation for protospace or adjacent motif. The long story short is that it has a particular sequence that it scans DNA for. In the case of CRISPR-Cas9, it's looking for this NGG sequence. So any nucleotide followed by 2 guanosines. It's just looking for that sequence. Once it finds that sequence on DNA, it will bind to the DNA molecule, and it will unravel it. It will pull the 2 strands apart. And then it looks for, it will compare one of those sequences to its guide sequence, and it just checks for complementarity. So, it just checks, do the base pairs between my guide sequence and this piece of DNA match? If they do match, then it triggers endonuclease activity, and it will do a double-stranded cut on the DNA. You can, and I say, people have modified this so that it only performs like a 1-stranded cut. People have modified this so like it doesn't cut at all, and it will just bind and just kind of sit there until something knocks it off or thermodynamics get to it. But that's the general gist. So what happens is so this CRISPR, like I said, this is part of the bacterial immune system. And so what it will do is when it identifies viral DNA, when it has run across viruses, it will incorporate some of that virus DNA into a particular portion of its DNA, and that will create these guide sequences. And then the CRISPR-Cas, so then the next time a virus or that same virus or a related virus injects its DNA into the cell, the CRISPR proteins will already be out looking for that viral DNA, and will just cut it up and make it completely useless before it has a chance to hurt the cell. So like that's how CRISPR like protein formed in the wild. So we use it for DNA editing, and or DNA gene silencing, and we do this through two different mechanisms. So after it makes this double-stranded cut, we can do non-homologous end joining. So what happens, and we just basically, in this instance, we basically just let the body's framework do what it wants. And the cell's response to seeing two flailing strands of DNA is to merge them together by whatever means necessary. And so it will just it triggers like a whole repair cycle, and it will just grab whatever nucleotides are available and put them together. So that's this non-homologous end joining, and that frequently leads to like these indel delete mutations, which if you target particular, suppose that you target like the promoter sequence of a particular gene, when it introduces these new nucleic acids, it messes with the promoter, and now you just like don't have, now this gene is turned off. So that's typically how we use this. Or alternatively, we can use what's called HDR, or homology directed repair. So in addition to like adding this CRISPR Cas9 protein, we'll include a strand, a complementary section of DNA that matches that matches part of the portion and then contains the correct gene sequence. And in which case, when this happens, instead of grabbing random nucleotides because it doesn't know what to do, it will see that this DNA is very similar to that DNA, and it will use this as a template to fill in that gap. So this allows you to like do like very specific gene editing at specific locations. Again, conceptually, I just want to explain like that's kind of how that works. We're not going to go into all of the nitty-gritty details about how either of those work. All right, so the CRISPR Cas13 system wants to do that same thing, except instead of targeting DNA, it wants to target RNA. So instead of looking for a like a particular PAM sequence on on DNA and like unwinding, it doesn't have to bother with that. It just looks for its particular sequence and then just checks for complementarity. And then because it's RNA, when it gets cut, it just gets marked for degradation. So you can use that. You could similarly to the Cas9 system, where you could silence genes. Here you could silence transcripts, which is just the same thing, just at a different spot. All right. So first they identified like this the capabilities of the Cas13 to target RNAs, and then they went about this selective characterization program to look for different variants of this protein in different bacterial cells. Different bacterial species, to find like the one that was like that performed best. So specifically, they found this Leptotricha wadi exhibited both high specificity and sensitivity. So then to test that, they created a plasmid that contained the gene sequence for the Leptotricha wadi Cas13A protein. They put it it into the E. coli. And then they included a gene sequence that includes the guide RNA that would be used to target like the particular genes. So so some of them were given one that targeted a particular gene, and then some of them were giving non-targeting, so like nonsense, like something that wouldn't connect at all. And then by comparing the amount of and then they grew that up on okay, that's not relevant actually. So they were growing this up on lactamase media, and they added this beta lactamase to make sure that the E. coli kept the plasmid, because it now has this beta lactamase to not die from the beta lactamase. So that's the idea for that section. So then we just compare the number of colonies for each gene that they targeted. So the idea being that if you turned off, we're going to target important genes, and if complete the transcript and that gene doesn't get transcribed, then, the RNA doesn't get translated, then there should be fewer colonies. And so what we can say and what we can see here is that, like in the non-targeting, it's pretty regularly 10 to the third number of with the targeting, we get, in the hundreds or tens of colonies in the best case scenarios, and then most of the time lower than in the non-targeting examples. So this illustrates sensitivity. This does work. They also did a PFS, which is just the protospacer locations, and they did positions 1 through 4, and they just checked like is there a, so this is a checking for preference at these locations for the four different nucleotides. So this is like a traditional like logo like analysis, and what we can see is that it doesn't, like there's not a strong preference for any given nucleotide at a particular position. So not only is it specific, but it's sensitive. In that it will perform, regardless of what these 4 locations are, it performs. So, all right, I know that was a lot. So any questions right now? Seeing nothing, hearing nothing, we'll just move on. I'm sorry, these images aren't the best. So they also checked if it could be say if it could be localized, say if it could be expressed and localized in mammalian cells. So they took the LWACAS13 gene. They also put on this GFP is green fluorescent protein, and they just put that on the say they just tacked that protein onto the end of the gene, and then in one instance they gave it a an NES tag, and an idea so nuclear export signal. That's what it is. So the idea here is that if this protein is produced, it will fluoresce green, and we'll be able to tell where it's at. And if it's on the if it has this NES signal, then it will not be. It will be exported from the nucleus. It will be produced. It will fluoresce, and it will not be in the nucleus. And when they did their fluorescence experiments, you can see like here are the the center, like that's where the nucleus is, and like we can see the GFP is outside of the nucleus. So great, it worked. Then we did the same thing here, except instead of giving it the NES, the neutral export signal, they gave it the NLS, which is the nuclear localization. There we go. Nuclear localization signal. And here again, we show that it stays inside, like it's it's clearly contained in like these little specific bowls, whereas this one's like more diffuse. So here we see that like they were in fact contained in the nucleus. So not only can it be expressed in mammalian cells, but it can be localized to either the cytoplasm or the nucleus of said cells. All right, so this was another set of experiments that they did. So here they used an NLS tagged LY-Cas13 system, and they showed that it can be more effective than simple shRNA for transcript knockdown. So we have two different same cell line. We give it two different plasmids. One that has this Cas13 system, one of them that has this shRNA system. And then we're going to give, and we'll transpose those into the same cell line, but two different splits. And then we're going to give them a different plasmid that contains this Gluck and Cluck genes. So therefore, we're not going to target any native genes with this. We're going to target these two other genes with it, so that we know that like we're not going to mess with anything else. Like we're not going to have any no conflating variables. This should be expressed, this should be expressed about the same across the two things, and then we'll just compare. If the system works, they'll degrade the RNA, and then we can measure the Gluck-Cluck ratio by independent readout. So both of these are also fluorescent proteins. So we just measure the fluorescence, which is what it means by like the normalized luciferase. So they also included a luciferase protein to use as the control. And then we can just compare the ratio between them. So here we have with the NES, with the NLS, and with the shRNA. So if it's in the nucleus, if it's contained in the nucleus, it's less effective. But if it's exported outside of the nucleus, it's at least just as effective, if not more effective than this shRNA. And that makes sense, right? Because where are most of the once a transcript is made, it gets exported into the cytoplasm to be translated, right? So it makes sense that if we export it from the nucleus, we'll get more activity. All right, so are there any questions about this part? Okay. I don't think we're gonna quite get through all of it. So let's just do this like this last one. So here, like what the ideal circumstances are for this. Like it wants to have a nuclear it wants to have an NES tag, a nuclear export signal. Now let's check like all of the contexts, like how well does this actually do? So they created a group, they created a list of target genes that they wanted to target. Then they tiled their guides across those gene links. So if the gene length is, so here we see like there, this gene is only, let's say 600 nucleotides long. So we know that we're going to take our guide sequence and we're just going to, place one here, and then we're going to move it some fixed length here, and we'll just tile it across the transcript. And then we will perform, and then we'll do this arrayed plasmid cloning so that each plasmid contains the same gene, will have a slightly different, guide associated with it. Then we'll do transfection of that system into our different cells, and then we will do some RNA sequencing, and we'll be able to compare the results of just by normalizing the counts. So in our control sample, there were X reads for this sample. In our guide experiment 1, there were Y reads. So we'll compute the ratio between them, and that will be like the normal, like the knockdown between them. Does that make sense? Okay. So then they just plotted this. This is the distribution for each guide at this position, like the average of the 3 replicates for that guide at this position. And what we can see is that there's for these 4 different genes, and what we can see is that there's an underlying pattern here. Some positions appear to be really that work really well, and some positions don't work well at all. Some of them work very well sometime. So like this one has like a really large. This one has this point has like a really large error bars. So some points work really well sometimes and not very well other times. So the idea here is that there's that the system will it works in a broad number of contexts, but there's some underlying things going on for it. Only, let's say, it has some type. There's some preference biologically why that it works in some areas and not in others. Like it doesn't work universally well in all contexts. All right, that's. So here they did some specific mutations in those guides, so that they had mismatches, and they show that it's fairly robust. That there's not that it is sensitive to mismatches, but it's still like so this is the non-targeting, but in each instance where it's still targeted, it will performed better than the control. So it's fairly robust, and it doesn't care too much about the mismatches. And then here they illustrated, they deactivated the endonuclease activity, so it wouldn't cut, and so instead it would bind. And they targeted two different sections. One of them, 2 different transcripts. One of them that was likely to stay in the cytoplasm, and one of them that was likely to be contained to a particular compartment. And they illustrated, so specifically these stress granules. So they illustrated that it will that it's capable of binding to transcripts and then following those staying bound to those transcripts as they are moved to different compartments of the cell. So you could, in theory, if you had like a really sensitive fluorescent setup, you could, in theory, track transcripts as they moved throughout a cell in real time using this Cas13 system. I understand explanations were not particularly elegant, but I thought we just wanted to go through for time. So is there any other questions?
If you don't have any questions, we'll wrap it up. But I really want to thank Xander for taking our time and starting this committee and starting this journey, the journal club journal event committee. We're going to start doing these journal events from now on, hopefully once per month. And as you can tell, there are places where we're completely lost, knowing what's going on, right? And so this is something that we want to improve on, and now we want to change that confused state to curious step where you start asking questions. And so for that, we want to start promoting the habit of reading research paper, right? So unfortunately for this event, we didn't send out the research paper. From now on in future, we will send out the research paper and highly recommend that you read it. And we will have an active discussion throughout the paper. So we'll have our paper presented in the first 30 minutes, and then the last 30 minutes is complete discussion. And so that is crucial, and we really want to emphasize that we ingrain this habit of reading research paper and having these questions as we read more and more. And as all the PIs and all these PhD students will be telling you that how much effort and time you put in is how much you're going to gain out of it, right? And that's kind of the goal of the entire idea of a journal committee here. And so I hope that you enjoyed this and learned how to read research paper, the basic ideas, and kind of got the idea and the gist of like how to cope with complicated text. Like in our case, this was CRISPR Cas9. Many of us may not have read or get exposed to this paper before and feel really confused. And we want to overcome this threshold and get to the stage where we are asking questions about this. Even though we're confused, we want to learn more instead of just passing out, right? So that's what we're trying to attain here. Again, thank you, Xander, for taking out time and presenting. Yes, we are officially done for our first journal event, and our next event is on next Thursday, 2 p.m. We'll be continuing our BioPython series, so it will be sequence alignment. We'll be going over. This event will be in person. We are having it in IT building 273. We will send out more information in upcoming week. We will hope to see you there. Any questions? If you don't have any questions, then we are officially done with the event. Thank you everyone for joining the event, and see you guys next time.